మా వాయిస్ వినిపిస్తుందా? ఓకే ఓకే. సో స్క్రీన్ కనిపిస్తుంది కదా? సో మనం నైట్రోజన్ బేసెస్ కోసం మాట్లాడుతున్నాము. సో వెన్ యూ అబ్జర్వ్ ద నైట్రోజన్ బేసెస్, సో A pairs with the T, that means adenine pairs with the thymine. Similarly, guanine pairs with the cytosine. So it is the condition you will find in the DNA molecule. Oh. But in case of RNA molecule, so in case of RNA molecule, so here instead of thymine you will find the uracil. Adenine pairs with the uracil molecule. Similarly, guanine pairs with the cytosine molecule. So here actually these are the hydrogen bonds. So double hydrogen bond, triple hydrogen. So in this case, A, G are known as the purines. Similarly, T, C are known as the pyrimidines. So here also Ag present, and that is purines. So C is a pyrimidine, and this U also pyrimidine. So these are the pyrimidines. Ag are the purines. So altogether, purines are A, G, adenine, guanine. Pyrimidines are thymine, uracil, cytosine. So let's observe the structure. So you know about the structure of benzene. So this is cyclohexane. If you write alternate double bonds, it becomes benzene. So every location you will find C is present, and remaining bonds are hydrogen bonds. So in this structure, at one location, if you replace this carbon with nitrogen, for example, if you replace this carbon with nitrogen, so as you know that nitrogen is able to form 3 bonds and carbon is able to form 4 bonds depending on their valence. So this molecule is known as the pyridine. But actually we require pyrimidine. So pyrimidine means, so in this structure of pyridine, if you place one more nitrogen over here, then it will become our required pyrimidine. So it is the structure of pyrimidine. Similarly, here we have purines. So for that pyrimidine structure, let's have the structure over here. So for that structure, if you attach a 5-membered ring with having 2 nitrogen atoms like this, so it will become purine. So this is a molecule of purine. So this is the basic structure of purine, and this is the basic structure of pyrimidine. So always remember that here the numbering is important. So this is the first position, second position, third position, fourth position, fifth position, sixth position. So this is the numbering for pyrimidine. Similarly, if you observe the purine, here the first atom is this one, first position, second position, third position, fourth position, fifth position, sixth position. So it is the anti-clockwise rotation, then clockwise, seventh position, eighth position, ninth position. So first we wrote in the anti-clockwise direction, 1, 2, 3, 4, 5, 6. Then second ring is 7, 8, 9, clockwise. And then anti-clockwise, clockwise. In that system, we will give the numbering. So here, this is the numbering pattern. You have to remember this. So coming to the first molecule, that is adenine. First molecule, that is adenine. So adenine is chemically it is 6-aminopurine. 6-aminopurine is nothing but adenine molecule. That means in this purine structure, so already we have the purine structure. Just if you add the amine group means NH2. So at what location you have to add? At the sixth position, you have to add amine group, NH2 group. It will become the purine. That means that is adenine. So let's have the structure once. So if you observe the numbering 1, 2, 3, 4, 5, 6, 7, 8, 9. So next task is placement of nitrogen atoms, and then here and here, here and then here. Next, to stabilize the bonds, so first bond, second bond, third bond, and here is the third one. So sixth position means this is the position, sixth position. So here you have to write NH2. So now this molecule is nothing but the adenine molecule. So similarly, you have one more purine, that is guanine. So one more purine is guanine. So guanine means 2-amino 6-oxopurine is nothing but guanine. 2-amino 6-oxopurine. That means firstly you need to draw the structure of purine, then placement of the molecules. So first you need to draw the structure first. So generally when you observe the structures, we get fear. But the thing is, if you properly learn, then it will becomes very easy. So next, placement of hydrogen bonds to compensate the 3 bonds for nitrogen and remaining bonds for the hydrogen. So whenever if valency is less, simply place the hydrogen atoms. So next, numbering. So first, second, third, fourth, fifth, sixth, seventh, eighth, ninth. So 2-amino 6-oxopurine. So we have the purine structure. So 2-amino means at the second location you have to place the amine group, NH2 group. So 6-oxo means at the sixth position you have to place the oxygen atom. So then it becomes guanine. 2-amino 6-oxopurine is nothing but guanine. So these are the 2 purine molecules. So these are the 2 purine molecules. So next we move on to the pyrimidines. So we have 3 pyrimidines: thymine, uracil, cytosine. So first we start with the uracil molecule. So uracil means So here I will write 2,4-dioxypyrimidine is nothing but uracil. 2,4-dioxypyrimidine. ఏంటమ్మా? కనిపించట్లేదా? Okay then. So uracil means 2,4-dioxypyrimidine. So next, thymine. Thymine means 5-methyl 2,4-dioxypyrimidine. The only difference is that at a 5' position, you will find a methyl group. So methyl group means CH3 group. And last one is that cytosine. So cytosine is 2-amino 2-oxo 4-amino pyrimidine. 2-oxo 4-amino pyrimidine. So let's draw the structures. So first we start with the uracil. So 2,4-dioxy. First you need to draw the basic structure. So here it is the basic structure. Nitrogen, nitrogen. So place the required bonding and then give numbering. So I told you that this is the first atom, second, third, fourth, fifth, sixth. So first molecule is 2,4-dioxypyrimidine uracil. So at the 2' location you have to place one oxygen. Similarly at the 4' location you have to place another oxygen. So this is the uracil molecule, 2,4-dioxypyrimidine. So next molecule is thymine. So I told you that only difference is methyl at the 5' position. Methyl at the 5' position. So first one, second one, third one, fourth one, fifth one, sixth one. So thymine means 5-methyl 2,4-dioxypyrimidine. That means same structure. 2,4-dioxy, 2,4-dioxy. But the thing is, at the 5' location, you have to draw methyl group. You have to add methyl group. CH3 group is methyl group. So this is the thymine. And last one is that cytosine. So 2-oxy 4-amino pyrimidine. So here we draw first one, second one, third one, fourth one, fifth one, sixth one. To compensate the double bonds, so here we have added the double bonds. So 2-oxy 4-amino, 2-oxy 4-amino NH2. Pyrimidine is nothing but the cytosine. So these are the structures of nitrogen bases. These are the structures of nitrogen bases. So we come across with the purines and then pyrimidines. So adenine means 6-amino purine. So purines possess 2 rings. Pyrimidines possess single ring. So first one is adenine. 6-amino purine is the adenine molecule. Second one is guanine. So here 2-amino 6-oxo purine. And coming to the pyrimidine molecules, first one is uracil, 2,4-dioxypyrimidine, and followed by thymine. The difference is at the 5' location you will find the methyl group. So here 5-methyl 2,4-dioxypyrimidine is the thymine, and last one is cytosine. So here 2-oxo 4-amino pyrimidine is cytosine. So like that we come across with the structures of nitrogen bases. Next, we need to see the DNA structure. What are the bonds are present? As you know that DNA is composed of, nucleic acid is composed of nucleotides. So more number of nucleotides, they are bonded with each other with the phosphodiester bond. And then you will find two helices in case of DNA, single helix in case of RNA. So just we are going to see only the single helix. So how it is going to form. So where you will find the what type of bonds it possesses. As you know that we require three things. So one is phosphate atom H3PO4. So in this place you can take PO4 3 minus also. So second one is that nitrogen base and then pentose sugar. So these are the three ingredients you need to draw. You required these three things are required to draw the structure of DNA molecule. Just we are going through the single helix structure only so that you can understand easily. So if we go for double helix, you may get bit confused. So that's why I'm taking only single helix structure. So first we will take the sugar moiety. So pentose sugar moiety. So DNA means 2' deoxy, right? So this is the first prime, 2', 3', 4' and then 5' CH2OH. So at every location you will find H and OH. This one is DNA. Obviously at the 2' location you will find absence of oxygen. H, OH, CH2OH, H. Coming to here H. So here actually at this location you will find the hydroxy molecule. But what happens is that nitrogen base is going to pick up this location. So hence for that purpose this hydroxy is replaced with the nitrogen base. So for my convenience I am taking a pyrimidine here. So I have drawn only the general structure. So this is the general structure. So coming to the phosphate atom. So here in between the sugar and nitrogen base, you are observing the bond. So this bond is known as the glycosidic bond. Why? Because whenever sugar is involved in the bond formation, obviously it will become glycosidic bond. But in between the nitrogen bases, so if this one is the guanine, obviously the opponent component, sorry, this one with the cytosine, obviously opponent component becomes the guanine. So there you will find the 3 hydrogen bonds like this. So I am not going to draw the structure over here. So actually this bonding is nothing but the hydrogen bonds. So at the CH2OH location, so always DNA synthesis or any nucleic acid synthesis takes place from 5' to 3' direction. 5' to 3' direction. So actually this is nucleoside it is. It is the sugar molecule and this was binded with the nitrogen base. So this is nucleoside it is. But actually for the formation of nucleic acid you need nucleotide. That means if you add the phosphate molecule. So here at the CH2OH location, here the phosphoric acid will come over here like this. So P double bond O, O minus, O minus, O minus. So here, in between these CH2OH and then PO4 3 minus, you will find the formation of bond here. Actually, it is phosphoester bond. It is. So you will find phosphoester bond. So at this location, you will find phosphoester bond like this. So in between the phosphate and sugar moiety, that bond is phosphoester bond. Phosphoester bond. In between the nitrogen bases, hydrogen bond. In between the sugar and then nitrogen base. So here you will find the glycosidic bond. So I need to pick up the color. So it was struck actually. So I will draw with this only. Okay, this one. So this is the one nucleotide. So this nucleotide forms a bond between another nucleotide. So another one I am picking over here. So first I have drawn the sugar moiety, CH2 phosphate also I am taking here, PO. It's not picking. Okay, I'm not. So here CH2OP. Here one more oxygen, O minus, O minus, H, 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 H. Here it is H. Here it is one more molecule of nitrogen base. So this is the another nucleotide. So in between these two here, in between OH and O, you will find the formation of new bond. So here what happens is that you will find a bond in between phosphate and hydroxyl molecule like this. So this bonding in future this bonding will become the phosphodiester bond. So previously in between the sugar and phosphate molecule, we have seen the single bond. That single bond actually it is phosphoester bond. But here at two locations, you are seeing the, so here at one location phosphoester bond, at this side also phosphoester bond. So this is the phosphoester bond, this is the phosphoester bond. Hence it is the backbone of the DNA is composed of phosphodiester bond. Phosphodiester bond. Like this, always from the 3' direction, always this 3' hydroxy molecule is involved in the extension of the DNA segment. So we have structures over here. So by observing those structures, you can get a clear-cut idea. So here it is. So I told you that. So 6-aminopurine. This is the 6-aminopurine. Sixth position it is. This is the ribose sugar moiety. And the 3 phosphates. Actually, this is the structure of ATP molecule. So as you know that when ATP molecule, adenosine triphosphate molecule, when it gets hydrolyzed, you are going to get ADP, adenosine diphosphate plus inorganic phosphate. If this ADP again going to get hydrolyzed, you are going to get AMP, adenosine monophosphate plus inorganic phosphate. So like this conversion is there. So here if you observe the ATP, the energy currency of a cell, adenosine triphosphate means 3 phosphate moieties are present. So this is the alpha, beta, gamma. So when you break down one phosphate molecule, you are going to get 7.6 kilocalories of energy. 7.6 kilocalories of energy. So here if you observe that this is the phosphate moiety and here is the ribose sugar molecule and here is the nitrogen base. So adenosine means ribose sugar plus nitrogen base, nucleoside it is. If you add single phosphate, nucleoside monophosphate. If you add 2 phosphates, nucleoside diphosphates. If you add 3 phosphate, the entire structure is nucleoside triphosphate. So actually it is adenosine triphosphate. So coming to the DNA, so here we have the DNA moiety. So if we observe, these are the phosphate molecules and here we have the sugar moieties. So this is the one helix and this is the another helix. So this is the this helix and this one is the this helix. So in between the steps are nothing but the nitrogen bases bonding. So A pairs with T with 2 hydrogen bonds, G pairs with C with 3 hydrogen bonds. So here in between nitrogen base and then phosphate molecule, this bond is the phosphoester bond it is. But this phosphate is making bonds with 2 sugar moieties at both sides. So this one also phosphoester bond. Hence it is phosphodiester bond. Phosphodiester bond. Coming to the in between the nitrogen bases, these are the hydrogen bonds. In between the sugar and then nitrogen base, you are going to observe the glycosidic bond. Glycosidic bond. So this is all about the nucleic acids. This is all about the nucleic acids. So you are going to get repetition like if adenine combines with sugar moiety, then it will becomes adenosine. If adenosine, when it added to the phosphate moiety, then it will becomes adenosine monophosphate. So like that you are going to get repetition. Guanine plus sugar, guanosine. Cytosine plus sugar, cytidine. Uracil plus sugar, uridine. Thymine plus sugar, thymidine. If you add single phosphate, guanosine monophosphate, cytidine monophosphate, uridine monophosphate, thymidine monophosphate. So like